to this webinar to discuss our phase 1 results for HMBD-002. I'm James Garner. I'm the Chief Executive of Percheron Therapeutics, and it's a pleasure to be your host today for this discussion. Some of you will be aware we've recently appointed a Chief Medical Officer of the company, Dr. Gene Kennedy, and he's actually still coming on board with us, which is why he's not with us today, but we're super excited to have him on board, and he's going to be instrumental in the future analysis and discussion of this work. But for today, I'm going to be talking through the results we've seen from our phase 1 study. This is our first data release for HMBD-002. And since we licensed the molecule about 3 months or so back, and I think it's a really valuable opportunity to try and give you some sense of why we're so excited about the drug, why we think this has been such a transformative step for Percheron, and why we think this has the potential to be a very meaningful, very important new therapy for cancer. So we're really excited to have this opportunity to share data with you today. Just a quick procedural note, I'm going to talk to some slides for about 20 to 30 minutes and try and give you an overview of the data that way. We will then open for questions, and we will we'll ask everybody to submit their questions via the Q&A box at the bottom of the screen. Those who've been on these Zoom webinars before will be aware that we don't have the ability to bring you directly into the conversation. So we ask you to submit your questions through that Q&A box. We may likely reword them or amalgamate similar questions. And if we don't have time to get to all of them, we'll have other forums to talk through these points over coming weeks, not least, of course, the company's upcoming annual general meeting on Wednesday, 22nd of October. I'd just say that today's slides have been announced to the ASX, so you can review this presentation at your leisure. You can download it from the ASX and access it that way. And we will try our best to make a recording of this presentation available after the fact so that you can review it in comfort afterwards. So with those introductory remarks out of the way, we'll jump straight into the presentation. So let's get started here, and just a little bit of recap first of all to remind everybody of the context of today's results discussion. We licensed HMBD-002 into Percheron back at the end of June of this year, so we've had the drug in our hands for about 3 or 4 months now. The drug was developed by Hummingbird Bioscience, a venture-backed private company based in Singapore. They've raised about $350 million plus US dollars from some leading venture capital funds. Very, very interesting oncology drug discovery outfit. They partnered the drug to us, and it's now our drug to take forward. And the trial that we're discussing today, this phase 1 study, was actually completed by Hummingbird. This isn't our trial per se. This is a trial that they completed during their custodianship of the drug. But because there's always a little bit of a lag in the analysis and the interpretation of this data, we're only now seeing the results of the trial some months after our transaction completed. I just want to remind everybody a little bit about HMBD-002. Some of these slides are drawn from our corporate presentation, but I'm really just providing them here for a brief recap and for those who are new to the story. And I want to mainly focus on situating 002 within the landscape of immuno-oncology therapies, which, as I've said on a number of occasions, one of the most exciting new areas of oncology drug development. This has really transformed the treatment of cancer over the last 25 years or so. As I've pointed out previously, some of the work underlying this was the basis of the 2018 Nobel Prize for Medicine. And what it really boils down to is a growing recognition that for tumors to establish themselves, for tumors to grow in a patient, they need to switch off the immune system around them. And they do that by virtue of protein molecules on the surface of the tumor cell. And you can see 4 of them shown here. There are others. This is just a select list. But tumors have to switch off the local immune system Otherwise, it will remove the tumor cells in the same way that it removes bacteria and viruses. And so drugs which disrupt that process and which therefore reactivate the immune system have been a very, very important new development in the treatment of cancer. And as you can see on the right-hand panel here, just 4 of those drugs account for almost $40 billion of annual sales, including one absolute titan of the field, Keytruda, the green bar on the right here, which is itself a 30-something billion annual product. This is the space in which our drug 002 is looking to enter. Now, our drug inhibits a different part of the process. It inhibits a target called VISTA, but conceptually, the idea is the same. We are looking to reactivate the immune system around the tumor so that the body's own natural defenses can remove tumor cells. Why VISTA? Why is this such a promising target? Well, as we pointed out as we were bringing the drug in a couple of months ago, VISTA is very widely expressed on cancer cells. It's a very broadly expressed target. So it gives us the potential to address this drug to a wide range of different cancer types. In general, the more VISTA you have on your cancer, the worse your prognosis. But particularly, the panel on the right here is really what intrigues us the most. When patients are or become resistant to existing immunotherapy drugs like Keytruda, the drug I just mentioned a moment ago, VISTA seems to be an important part of that resistance mechanism. So for all these reasons, this is an incredibly attractive target for us to try and develop a medicine against. Just going to jump ahead here and show you just a flavor of the preclinical data that got us excited when we were doing our due diligence for this molecule. 002 has been put into a wide range of animal models of cancer. And as you can see here, this is just a flavor of the data that we have, but very consistently we see significant and substantial reduction in tumor growth in mouse models of cancer. And in particular, in the panel you see on the right here, when the drug is given in combination with Keytruda, pembrolizumab, we see in this particular mouse model, at least, an almost complete shutdown of tumor growth. So this is the kind of data that gets us excited about a new medicine. Now, just to qualify this, mouse models are not always predictive of what we see in patients, but it is true that a cancer drug which doesn't show any activity in these kind of models is rarely efficacious in patients. So this is a really important screen for us as we're looking to take a drug forward. Finally, by way of background, I just want to stress that what we're talking about today is a phase 1 clinical trial. It is specifically a first-in-human clinical trial. That is to say, this is the very, very first time that this drug, HMBD-002, has been put into human patients. And that situates it roughly in the middle of the very, very long drug development process that many people on the webinar will be familiar with. As you can see here from the diagram on the left, out of every 10,000 or so drugs which start out in the lab, only about 5 or so make it to initiating human trials, and of those, about 20% become a marketed product. So we have now completed phase 1 human trials. That's the first step in the development of a new medicine in the clinic. And the success rate for cancer drugs going to phase 1 is about 50%. So we have already cleared, in some ways, the most challenging bar in the development of a new cancer medicine. We've been through all that preclinical work. We've now been through a phase 1 human trial. However, phase 1 trials are always safety studies. They are about learning how to dose the drug, about learning its most common toxicities. They're not designed to show efficacy. And indeed, when we do these kinds of studies in most diseases, we actually do them in healthy volunteers. For various reasons, we tend to do phase 1 studies for new cancer drugs in patients, but that is the exception rather than the rule. Because of that exception, we do sometimes get little hints of efficacy in phase 1, but stress again, this is not an efficacy study. It is not designed to answer the question whether the drug works. That will be the task of phase 2, and we'll touch on that a little bit at the end of the webinar. So with that background briefly reviewed, I want to go and just talk a little bit about the design of this study, just to give you a flavor of how it works mechanically, and talk a little bit, first of all, about the patients that were recruited. And as you can see here, this was a study that was performed in patients with advanced cancer. And specifically, the language used is advanced cancer patients with no available treatment options known to confer clinical benefit. What that means in layman's terms is that these were very, very late stage patients. These were patients who, to put it very directly, had run out of treatment options and were in the very, very final stages of their disease. That is absolutely common for these phase 1 first-in-human studies. For all kinds of reasons, we tend to do these studies in very, very late stage patients. But in a certain sense, it does set a very, very high hurdle for all our drugs, because it means that we're giving these drugs to not only very sick patients, but patients who are very advanced in the disease and tend to be very treatment resistant. So in some ways, this is the toughest possible way to examine a new cancer medicine. And it's important to bear that in mind as we discuss the results. Now, as the middle part of this slide shows, the design of this study was a very, very common design, what we call a multiple ascending dose study. And the way this works is that we start off giving a very, very, very low dose of the drug to just 3 patients. As you can see here, we started in this study with 20 milligrams of the drug, which is almost a homeopathic dose. But we're really doing that just to make sure we understand any sort of severe toxicities that result from administration. So this is very, very much in the vein of understanding the safety of the drug. We follow those patients for, in this case, 3 weeks. And if we don't see any significant toxicities, what we call dose-limiting toxicities, we recruit another 3 patients and we give them a slightly higher dose of the drug, in this case, 60 milligrams. Now, as you might notice, there are some of these groups where we have more than 3 patients. That typically means that some patients just were so ill that they couldn't remain in the study long enough to be evaluated. But so those patients had to get replaced. That's why we have 4 and 5, for example, in these first 2 cohorts. But we continue doing this, recruiting 3 patients at a time, each time increasing the dose. And our usual approach is to do this until we see 2 patients out of 6 get a dose-limiting toxicity. And at that point, we declare the dose below that as the maximum tolerated dose. And I just go over that design a little bit because it's important to understand why there are so many doses, but also we saw some interesting things in this particular study here. One other aspect of the study design here that's important to stress is that the study initially gave patients just HMBD-002. So the peach boxes on this diagram relate to patients who only had 002. But what the study then did was to recruit a further 4 cohorts who had 002 in combination with pembrolizumab, aka Keytruda. That was the $30 billion product I mentioned earlier. Keytruda is such a ubiquitously used drug in many of these cancers. We really wanted to understand at an early stage the potential to combine these 2 medicines. So this study gave us data both with 002 as a monotherapy and in combination with Keytruda, which is very, very useful data for us. A final point here, as you can see on the right, this study was conducted at really some of the leading cancer hospitals in the United States here. Stanford Cancer Institute, MD Anderson, Cedars-Sinai. These are absolute top-notch cancer centers. And so the studies bequeathed us not just some useful data, but I think some very enthusiastic champions for the product. And we're already in contact with many of these clinicians, and we'll be continuing to talk to them in the weeks and months ahead. So let's talk a little bit about the patients that were recruited. As you can see, the study recruited a total of 48 patients. 28 of them had 002 as a monotherapy by itself. 20 of them had it in combination with Keytruda, pembrolizumab. These patients were in their 50s on average, but quite a wide range. Youngest patient in the study was just 27 years old, which is sometimes a reminder, if it's ever needed, that cancer is a disease that can affect anybody, and certainly not exclusively a disease of older people. You can see here the broad demographics of the study. I won't go through these. I just point out here a couple of data points. The time since initial diagnosis, on average, a little over 4 years. These are patients who had been going through their journey with cancer for more than 4 years on average. So again, very, very late stage patients. All of these patients had metastatic disease. That is to say, the cancer had spread out of its original organs. So, for example, lung cancer, we sometimes see spread to bone or to liver or to brain. That spread is often the point at which cancer becomes, in effect, incurable. And so these patients were all in that position of having metastatic disease, and on average, they had had metastatic cancer for over 2.5 years. So again, just to hammer the point home, very, very late stage patients. On the left-hand side here, you can see a breakdown of the tumor types that we had in the study. Now, as is usual for these phase 1 studies, this is what we call an all-comers study. So we had patients with just about any kind of cancer who were eligible for the study. As it turned out, in the monotherapy arm, they broke down roughly in 20% blocks: pancreatic cancer, colorectal cancer, bowel cancer, breast cancer, squamous cell carcinoma, which is a bit of a mixed group, and then others. It's not deliberate that they break down so evenly. It just happened to be a feature of this study. In the combination group, about half of the patients had triple negative breast cancer. We'll talk a little bit more about what that is in a moment. And again, this really just reflects to some extent the specialist interests of some of the centers we worked at, and to some extent where pembrolizumab Keytruda is most commonly used. A point of interest on the right here, a little bit esoteric, but we did look at the tumor mutational burden for some of these patients. This test isn't routinely done for all patients. We only had this data for about 18 out of the 48 patients. But it's interesting because it's generally considered that patients with a higher tumor mutational burden are more responsive to immunotherapies, such as Keytruda, such as 002. And as you can see here, we had most of our patients in a more intermediate grade. So this is something we'll be really looking into in detail and discussing with clinicians, of whether this is an opportunity to enrich future clinical trials for more responsive patients. But we certainly didn't have in this study we certainly didn't have a weighting towards patients who have been more likely to respond, if I can put it that way. Prior treatment, these were, needless to say, very heavily pre-treated patients. These patients had been on multiple rounds of prior therapy. As you can see here in the monotherapy group, patients had had an average of 4 prior lines of treatment, and in the combination therapy group, an average of 5 prior lines of treatment. One patient, as you can see here, had had an astonishing 13 prior lines of treatment. That is to say, that patient had tried 13 different drug treatment regimens and had failed all of them, or at least had completed all of them, or had them all limited by toxicity. I think that's the most heavily pretreated patient I've ever seen in a clinical trial. So really, again, gives you a sense these patients have been through all of the treatments available to them. And why is that relevant? Well, we do know that the more heavily pretreated a patient is, the less the poorer their prognosis, and the less likely they are to respond to future treatments. So on the left-hand panel of this slide, you can see some data that shows median overall survival, that is to say life expectancy, for patients who've had 4 or 5 prior lines of treatment. As you can see, for a disease like breast cancer and lung cancer, it's perhaps around 10 months. For patients at this stage of the disease with pancreatic cancer, sad to say, it's measured really more in weeks than in months. And on the right, you can see here, it's not a fantastically clear slide, but we've reproduced it from a published paper. With each successive line of treatment, the progression-free survival, that is to say, the time before the patient's disease worsens, reduces. So we were getting patients at the right-hand end of that chart. So very heavily pretreated patients. We would expect them to have a poorer prognosis and to be treatment resistant. And so again, I want to just provide that context to give you a sense of the patient groups that came into this study. So let's talk first about safety. As I say, this is a safety study first and foremost. This is the data that is of most interest to regulators like FDA. But actually, there's very little to talk about because the reality is that 002 was an extremely safe and well-tolerated therapy. You can see here on the left-hand side of this slide, bars showing what we call treatment-related adverse events. That is to say, toxicities that were attributed by the clinicians to the drug. So if a patient goes and stubs their toe, that can be an adverse event, but it's not treatment related, it's an accident. We're interested here in things that are treatment related. And we're particularly interested in grade 3 and above. We rate toxicities on a 5-point scale in cancer. Grade 1 and 2 are very mild. Grade 3 and 4 are more serious, and grade 5 are fatal toxicities. Thankfully, we did not have any of those in this study. But as you can see here, we had a handful of patients in both groups having grade 3 and above treatment-related adverse events. By and large, these were very standard things: nausea, diarrhea, fatigue, rash. I should say that these are also common side effects of cancer. So understanding how much of this is due to 002 and how much is due to the disease is never clear in these studies. We err on the side of caution, but it's only in later randomized controlled trials that we really understand how much is, if you like, the background and how much is the drug. I mentioned earlier that we typically escalate the dose until we see 2 dose-limiting toxicities. We did not do that in this study because in the entire study, we only saw 1 dose-limiting toxicity. Again, this is a first for me in my experience of cancer drug development, but this study was, in a technical sense, unable to establish a maximum tolerated dose because there were so few dose-limiting toxicities. So they went up to a dose of 1,400 milligrams, and at that dose, we think it's probably maxing out the potential efficacy of the drugs. They didn't go any further, but really a very, very clean safety profile for a drug used in advanced cancer. In particular, one point at the bottom here, which is so important, it's got a little yellow highlight on it. We did not see any cases of cytokine release syndrome. And I'll touch on that in a moment, but cytokine release syndrome is a very, very nasty phenomenon where the immune system is overactivated. And it's an occupational hazard with any drug like this, and it has bedeviled other drugs that have been developed against VISTA. It has been a real challenge for drugs in this class of medicines. We'll talk about that in a moment. We did not see a single case. We didn't even see any hints of it in this study. So that's really, really encouraging. Time on treatment, these were very, very sick patients. So by and large, they did not remain on drug for very long. As you can see here, the average patient was on treatment for about 2 months. And so again, as we're interpreting the data, we have to recognize that the majority of patients had a relatively short period of exposure to 002. Again, that's very common for these phase 1 studies, but it just gives us, a sort of caveat around the data as we look through it. And then finally, I just want to touch a bit more deeply on this point about cytokine release syndrome. Again, this slide is drawn from our corporate deck. But as people who've been following the story for the past few months will be aware, a number of companies have been trying to develop drugs against VISTA. It is a promising target in the treatment of cancer. We are not alone in this space. But all of the other drugs are of a particular type called IgG1 antibodies. I won't go into details today about what that means, but it is a point of differentiation for 002. It is an IgG4 antibody. And it is that by design, Hummingbird hypothesized that an IgG4 antibody would cause less risk of cytokine release syndrome. And certainly, if you look at these IgG1 antibodies in the hands of companies like Janssen, part of Johnson & Johnson, and so on, many of those drugs have been limited in their development. They have been abandoned during their development. And we believe that cytokine release syndrome has been one of the key challenges behind those. So the fact that we have not seen a single case with 002 is very relevant and very encouraging. I'm going to move now to talk about efficacy, which is always the thing we get most excited about in cancer trials, rightly or wrongly. I just stress again, this is a safety study, so all we can ever see in these studies is hints and glimpses and suggestions of efficacy. But nevertheless, it was one of the questions we were most interested to explore. And so let me start by talking about changes in tumor size, or what we in cancer drug development call overall response rate. Uh, Tumors do not usually shrink of their own accord. So if we see tumor shrinkage, we usually ascribe that to benefit from the drug. And in fact, tumors usually don't remain the same size of their own accord. One of the definitional characteristics of cancer is that it is uncontrolled growth. And so even if we see what we call disease stabilization, that is, the tumor remains the same size, we tend to attribute that to some benefit from the drug. So when we look at a drug like this, one of the things we often look for is what is happening to the size of the tumor. As I say, we refer to that as overall response rate. If a tumor completely disappears, if it is no longer detectable on an MRI scan or a CT scan, we refer to that as a complete response. And obviously, very encouraging when we see that. If a tumor shrinks more than 30%, we consider that a partial response. If it stays between a 30% shrinkage and a 20% growth, we consider that stable disease. Anything greater than 20% growth or 20% increase in size is considered progressive disease. And we usually consider that to signify failure of the treatment. But in fact, there's a lot of discussion about this in oncology circles. We think maybe that's a pessimistic way to look at this data. But nevertheless, here is the overall response rate data for 002. And as you can see, between 18 and 30% of patients had stable disease. That is to say, their tumors for some period of time did not grow more than 20%. The rest of the patients either had progressive disease or were not evaluable. Bear in mind, this is data from the entire study. So this includes many of those early patients who had very, very tiny doses of the drugs, in some cases for very short periods of time. So this is sort of all in data. Now, how do we interpret this? And I'm conscious in discussing this that there are a number of ASX-listed biotech companies recently who have been breathlessly announcing to ASX almost on a weekly basis that they've just had one more partial response in a clinical trial or whatever the case may be. And so I want to talk investors through a little bit how we look at this data and what this should mean. And the best way is to compare this against what we see with other drugs. And in particular, I want to address the question of whether it's a disappointment that we haven't got any partial responses in here. And the short answer is no. If we look at some comparable drugs here, just for illustration, you can see the left-hand bar in this slide is the data from 002. We've merged the monotherapy and combination therapy groups together for simplicity. Now, the next bar along is a drug called Nexivar, sorafenib. It's been around for about 15 years now, but it revolutionized the treatment of kidney cancer and liver cancer. As you can see, the two bars look almost identical. Nexivar was a $1 billion drug at peak sales. It was making more than $1 billion a year in its heyday. The next bar along is Yervoy. It's another immuno-oncology drug, couple of responders there, but otherwise really quite similar data to our drug. And what this reflects, I think, is that many of these immuno-oncology drugs are really about limiting the growth of a tumor rather than melting it away in the way that chemotherapy used to do. Avastin famously had no response in its phase 1 study. It went on to become for a long time the most successful cancer drug in history until it was knocked off its perch by Herceptin. And then again, just to make the point a bit more, Erbitux, you see at the right here, another very successful drug with broadly similar sort of profile in phase 1. So our results here are not in any sense any kind of negative reflection on a drug. They look really a lot like other similar drugs at phase 1. And in fact, when we move forward, it is not usually on response rate that FDA approves new medicines. FDA usually likes to look for more time-based endpoints like progression-free survival and overall survival. And so as we think about the path to registration for this drug, we use this as a way of getting a feel for it, but ultimately it is those endpoints that are more material and more important. So let's look a little bit about the time course here. This is quite a confusing slide, and just for simplicity, we've removed some of the very low doses here, 20, 60, and 120 milligrams. They're so low, we think they really, in the end, give us limited useful information. But what you can see here is the change in tumor size, which is on the vertical axis, over time, which is the horizontal axis. And as you can see, for patients who received 180 milligrams of monotherapy, they progress very quickly. The tumor grows quite fast, and these patients don't remain in the study for very long. We think we can conclude that there's probably limited efficacy benefit from that dose. But as we move up, as we get to 360 milligrams and doses beyond that, you can certainly get a crude sense from this chart that we see patients remaining on treatment for longer, at least in some cases, and remaining in this important band between -30 and +20 percentage points. That is to say, remaining stable. And so this is broadly encouraging data. This gives us a feel for some sort of very, very crude dose-response relationship with the drug. It gives us a sense that at least some of these patients are seeing significantly prolonged disease stabilization. And when we look at the same data for the combination group, again, it takes a little bit of time to really digest this slide, and I encourage people to download it from the website and study it more carefully. But you get a similar sort of sense. Again, 180 milligrams, probably not a particularly useful dose going forward. But as we move the dose up, you get a sense that these lines flatten. These patients their tumors are growing more slowly, remaining stable for prolonged periods of time. And so we think this is a broad sense that the drug is providing benefit to some of these patients. Nevertheless, it's always hard to generalize. And as I say, every single one of these lines represents a patient with a different kind of tumor. So this is by no means comparing like with like. And actually, sometimes the most useful data in a phase 1 study consists in looking at some of the individual patients. So we have done that. We've been through each of the 48 patients in the study. And I picked out just a few to share with you today. There are others we could put in here, but we didn't want to give you a presentation of 8 or 10 different patients. So we picked out 5 case studies here. The first patient to talk about here, as you can see, 27-year-old male with metastatic dedifferentiated liposarcoma. As I mentioned earlier, sadly, a very young man afflicted with this very, very challenging disease. Liposarcoma is, to put it very crudely, a cancer of fat cells. They're a rare tumor, about 700 cases like this a year in the United States. And as you can see, median survival for patients with this disease is somewhere in the ballpark of 5 years. This gentleman had already been something like 7 years into his disease when he came on the study, had been treated with other drugs, including immunotherapy. So this patient had already had nivolumab and ipilimumab, trade names Opdivo and Yervoy, already had these drugs for 2 months with no real benefit. Came into our study, had 360 milligrams of 002 just as monotherapy, and remained stable for more than 1 year. And that is exceptionally unusual for patients at this very late stage of a tumor such as this. So a very, very encouraging signal that this patient has, in all likelihood, experienced some treatment benefit from the drug. 1 year on a cancer drug for a patient with any cancer at this stage is exceptional. So certainly this is one of a number of data points that gives us some feel for the potential activity of the drug. Another example here, a 67-year-old lady with triple-negative breast cancer. Triple-negative breast cancer accounts for something like 15 to 20% of all breast cancer cases. We call it triple-negative because patients don't have estrogen receptors, progesterone receptors, or HER2 receptors on the tumor. And those three kinds of receptors are some of the most common targets for cancer drugs. So what it means is that patients with triple negative breast cancer, or TNBC, are exceptionally hard to treat. These are this is considered a very aggressive form of breast cancer. It's a very difficult to treat breast cancer. It's not responsive to many of our common therapies. And this patient, as you can see, remained on drug for about 6 months with stable disease. Again, a patient who had had prior treatment with immunotherapy. They previously had 11 months on pembrolizumab, which had eventually failed. So again, a very late stage patient, already failed on a different kind of immunotherapy, 6 months of stable disease with 002. Again, a very encouraging signal for us. Third case here, a 49-year-old male with metastatic non-small cell lung cancer, lung adenocarcinoma. Lung cancer, of course, the most common cancer in the world. And remains an enormous healthcare challenge. And as you can see on the right, very heavily pretreated patient and had not just one, but in fact 2 different rounds of immunotherapy treatment with both pembrolizumab and with nivolumab and ipilimumab. So with Keytruda, and then later with Opdivo and Yervoy. And again, a patient who remained on treatment with 002 for a little over 6 months. And this was a patient who had our drug with pembrolizumab. Now, this was somebody who had already previously failed pembrolizumab, but when we gave them 002, they remained stable for this prolonged period of time, despite having very late stage, very aggressive lung cancer. So again, a really nice indication that our drug is providing some sort of benefit. Fourth example here, this was another patient with triple negative breast cancer, and really caught our eye as we saw very quickly with this patient quite a significant reduction in their tumor size. I mentioned earlier, cancers don't get smaller by themselves. This patient saw, roughly speaking, a 27% decrease in the size of her tumor. And that's very promising. Very, very near miss, because as you recall, a 30% decrease is enough for a partial response. So this patient, one almost feels if they'd remained on study for a little bit longer, might have crossed that threshold and given us our first partial response with the drug. As it happened, their tumor worsened elsewhere, and so they came off the study quite quickly. But again, a really promising glimpse of potential clinical benefit. And then finally, I think you sort of starting to get the sense here, but a patient with metastatic squamous cell carcinoma of the head and neck, and 59-year-old male. This was a patient who only remained on treatment for about 3 or 4 months, but again had quite a material reduction in the size of their tumor, about a 13% reduction in the size of their tumor. So again, it just gives us a feel for the sense that this drug is doing something. And not only doing something, but doing something across a very, very broad range of patients. We've just discussed lung cancer patients, breast cancer patients, head and neck cancer patients. And so we get a sense of this drug having potentially a very broad range of activity across different cancer types. So I'd just like to tie things together and talk a little bit about where we go from here. I'd be delighted to open for questions. And just a reminder for everybody on the call, you can ask questions using the Q&A box at the bottom of the screen, and we'll try and get through as many of those as we can. But to summarize the conclusions of this study, I think the key point here is that our drug seems very safe and well tolerated. It seems in many ways almost extraordinarily so for a drug used in advanced cancer. And that is the key objective of this study, is to understand its safety profile. And I think it is fair to say 002 passes that with flying colors. As we've noted, a very, very late stage population of very, very heavily pretreated patients. Nevertheless, we do see some very, very intriguing glimpses of potential efficacy here. We'll be continuing to study this data, really trying to pick apart, what might separate some of the more responsive patients from the less responsive patients. But nevertheless, this gives us a real feel that our drug is, in a sense, pharmacologically active. It is moving the needle. It is having an impact on patients. And that is really as much as one can ever hope for at this stage of development. And I think we're very encouraged by the data we've seen. And so the next step for us now is phase 2. And again, this is a slide out of our corporate deck. It's something we've started to discuss with folks, that we're in the process of thinking very, very carefully about what that phase 2 study will look like. We're out there talking to experts, to clinicians, to researchers to take their views on it. We have been very, very carefully mining the data from the phase 1 study. And we're looking forward to sharing some of our plans with you in the not too distant future. I think we recognize this as a question of very, very high interest to investors. And so hopefully within a short space of time, we will be able to share with you some more detailed plans about what we're hoping to do in phase 2. We've said we'll do that in the fourth quarter of this year, and I think we should comfortably be able to give you some sense of the path forward. So to summarize, here's where we are. You can see here some of the company's key milestones. Top of the list there, release of full phase 1 data. We are here. That's what we're talking about today. But in the background, we've been doing quite a lot of work about preparing for phase 2. One of the things that's happening quietly in the background is that work is underway in preparation to manufacture a new batch of drug for the phase 2 study. Again, those who are veteran biotech investors will know this is often a delicate point, and it can be quite a time-consuming point. So we're ahead of the game there. We've got things moving in that domain. You will have seen over the past couple of months some very interesting new preclinical data was published. Some from a team at Stanford University in Palo Alto, California, some from a team at the University of Texas Southwestern in Dallas, Texas. Really showing some very, very promising data for our drug in head and neck cancer and triple-negative breast cancer, respectively. So again, this is part of the data that we'll be studying as we think about phase 2. We're currently talking to clinicians about this, and somewhere in the fourth quarter, we will share with you our plans, and we'll look forward to talking those through in some detail with you. And that will line us up to hopefully commence phase 2 clinical trials of this drug in calendar 2026. Phase 2 studies are efficacy studies. It's where the rubber hits the road for a drug in development. So I think this is going to be a very, very interesting year ahead for Percheron. And with that, I'm going to conclude the webinar and be delighted to address any questions. I'm going to stop the screen share there and look forward to addressing people's questions. Just give us a second here to bring up some questions on screen. Let's see what we've got. So we've got a couple of questions about our license agreement with Hummingbird to start with. Given these apparently positive results, why did Hummingbird license the drug to Percheron? And can you please provide further detail on the financial arrangements? Let me talk to both of those points here. So first thing I should say is Hummingbird is a private company, but a very successful. As I mentioned earlier, it has raised some hundreds of millions of US dollars from some of the world's leading venture capital funds. But they are primarily a shop focused on the discovery of new medicines rather than on their development through clinical trials. And so I think part of their business model, so to speak, it's generally to sort of partner these kinds of medicines to more development-focused companies with a stronger clinical focus, so that they can focus on the real kind of groundbreaking science work. And this is a common model in drug development. We see a lot of companies that take a similar view. I should also say that this is a company that is focusing on some very, very niche technology areas, some very novel technology areas, antibody-drug conjugates, bispecific antibodies, T-cell engagers. They're trying to position themselves as a world leader in some of these very, very particular technology spaces. And so 002 is perhaps a slightly less consistent with that strategy. In terms of the financial arrangements, there's a fair bit of disclosure in our announcement at the end of June. We pay $3 million in the upfront for the drug, and then some contingent milestones and royalties, which are linked to its success, primarily to its commercial success, I should say. So Our financial obligations through the development of the drug are not onerous. Fundamentally, Hummingbird will make money out of this deal if and when the drug becomes a successful commercial product. And we like that because it means we can focus our resources on the development work, and it means that we have an alignment of interests. And they have continued to be, in fact, very, very supportive, very collegiate partners over these last few months. So I've got a next question here. Why did the dosing stop at 1,400 milligrams, 1,400 milligrams, instead of continuing higher to see if there was more efficacy? And it's a very good question. The rationale for this is that we know from a lot of computer modeling what we think is likely to be the therapeutic range for this drug. As I mentioned at the beginning of the presentation, the drug works by blocking VISTA. And as you can imagine, there's a point where you've effectively blocked all the VISTA that's available, and giving more of the drug is not likely to produce extra efficacy. Somewhere around 1,400 milligrams is probably something like that threshold. And so we wouldn't expect there to be a great deal of additional efficacy beyond 1,400 milligrams. We might start to see some toxicity if we continue to move it up, but it usually doesn't make sense in these kinds of trials to go beyond the point where we would expect to see efficacy. So Hummingbird, quite sensibly, I think, reached that point and stopped going further because they thought, well, we're giving as much of the drug as we could possibly need. I should say, just again, to reiterate, this is an unusual situation for a cancer drug. We are very often limited by safety in the development of new cancer medicines. So when we're at a point where we're even thinking about the therapeutic range in a phase 1 study, it's a very, very nice place to be. But a question here, do you plan to discuss these results with FDA? We absolutely will. The drug has an open IND application with FDA. Now, an IND, an investigational new drug application, that is essentially FDA's permission to run clinical trials in the United States. Veteran investors at Percheron will remember this was sometimes a challenging point with our previous program. This drug has not been so limited. This trial was conducted entirely in the United States under the oversight of the FDA. We will certainly be submitting the final data to FDA. We'll be sharing all the full results of this trial with them. And then we will likely, over coming months, be talking to them about path forward in various contexts. So we always like to develop drugs like this very much in lockstep with FDA, because they are ultimately the people who our data needs to convince. So short answer, yes, we will be knocking on the FDA's door. Right now is probably not the time. Some people on the call will be aware there's a federal government shutdown in effect. We usually see the effects of these things as being short-lived, but it certainly has some implications on organizations like the FDA in the short term. We have one more question here, which I'll address, and then we'll aim to wrap up the webinar here. And it is this: if the results continue to look positive, what is the potential value of HMBD-002? And look, that's a very difficult question to answer, because it depends very much on exactly how we move forward and which particular cancer types we put this drug into, how we situate it in relation to other therapies. And frankly, it depends on how well it works in phase 2 and other trials. But I would say this, that the average for a new cancer drug, depending a little bit on the data you look at, is that they tend to average something like about $1.6 billion in peak sales. That's to say, when they reach their maximum, when they're fully penetrated into the market, the average new cancer drug sells about $1.6 billion per annum in sales. And it usually takes about 3 or 4 years to get there. So it ramps up over that time. Some countries like the United States, it tends to ramp up quickly. Others, it's a bit more slow. But within about 4-ish years of launch, the average new cancer drug, that is their sales. And indeed, about 85% of cancer drugs that have been launched in the last decade or so sell for greater than $100,000 per patient year of treatment. So fundamentally, the commercial opportunity here, whatever cancer we go into, is enormous. Now, obviously, we have to get there. We have to generate the phase 2 data that shows the drug works. But there is, in almost any cancer we choose to pursue, an enormous addressable market. Having said that, the question was not specifically about financial value. And I do just want to maybe make one final reflection. It was motivated a little bit by a conversation I had with one of our shareholders in recent days. Cancer remains the most prevalent healthcare challenge in the Western world. 1 in 2 people will receive a cancer diagnosis in their lifetime. And this is still something where there is enormous unmet medical need. And so I think part of the value of a drug like HMBD-002 is the opportunity to bring new hope and new opportunities, new options to patients who are facing the challenge of a cancer diagnosis. So I think that is also part of the business that we're in at Percheron, and I think it's maybe a good final note to touch on in conclusion to this webinar. So, ladies and gentlemen, with that, I'm going to conclude our webinar today. I'd just say finally a reminder that the materials we've reviewed today are available through the Percheron website, have been announced to the Australian Securities Exchange. We will try to make this webinar recording available in coming days for you to review. And if you have further questions, please feel free to reach out to us at any time, info@percherontx.com. We're always happy to discuss with shareholders at any time. So we are here and available as you need us. And we'll look forward in the near future to sharing with you some detailed plans about our phase 2 trial. And as I say, I think it points to a very exciting calendar 26 ahead for the company. Ladies and gentlemen, thank you very much indeed.